All right, good morning, everybody. Happy Friday. I want to start off looking at the packback stuff real quick, just to show you a little bit of stuff. Right now, I have it, if I can make this work. I have it switched over into presentation mode, so all of this stuff I guess fits with FERPA or whatever. None of your names, although I was reading through some of the posts and there are maybe names mentioned in a response. You might see some of that. I did not look at that in presenter mode. But whoever wrote this, you don't need to spout off right now, but today you're just a curious mind. And this is me, a curious mind as well. But if I didn't have it on presenter mode, it would be me with my my little avatar, which is kind of hairy like myself. I don't know if you see my avatar when you do this or not, but I was like, oh, I kind of like that. I couldn't give him a ponytail. I couldn't quite figure that out. Not that made it look like what I thought I looked like, but I don't know. So I logged on to this before the semester started. Nobody was on here. And I looked at it the littlest bit, and I did not get back on here until this morning. I was going to do some stuff at my house, and I left my notebook that I had all sorts of stuff written down in, and my password was on that. And I thought I remembered it, but I didn't, not until I got to school. So I spent a little bit of time this morning going through this, and I can tell you, I am not done. I was thoroughly impressed with these postings. There is a lot of cool stuff, a huge amount of curiosity. There's all sorts of cool things that I started scrolling through. Some yet to have responses to them. I assume many of these will wind up getting somebody making some comments. So if you have not yet got on here, I highly recommend you do and start moving through here. I think I have virtually everybody signed up with this. I think maybe 10 or 12 yet to get on here, but pretty close to everybody. I think there were 80 posts that I saw. So there are lots of things, and I don't want to go into great detail this morning. Like I said, I haven't spent a huge amount of time looking through all of this, but I do want to just show you some of this stuff as your responses get in here. There are specific curiosity points. It mentioned some of this stuff before that you start getting as you're asking these questions, and dependent upon how they are asked. I believe your target is to hit 50 or above, all right, with that. And if it's not, then you need to start thinking about what's up. If you have been flagged for some of these questions, see some of the details about that as far as maybe how your questions are being worded. Things like that might be some of the possibilities. With that said, I do have a meeting with these guys next week to talk about some stuff. So as we are doing stuff next week, my meeting's Friday afternoon with them. So if there are comments or anything like that, I will try to address that with them next time. So if you got questions or anything like that, in the meantime, let me know and I will try to ensure that I get that stuff dealt with. So there were all sorts of interesting posts, all right, and definitely things that I can see you guys are thinking about microbiology. There's definitely some curiosity, questions about benefits, differences, probiotics. There's numerous things about probiotics, handfuls of stuff about essential oils, things about microbiome, and impacts with, should we feed our good guys? Why are we so concerned about the bad guys? Or what does our gut health have to do with things like maybe depression? All sorts of cool stuff, all right? And I did not respond to anybody, but I definitely will start making some comments. Don't be saddened if I don't comment on all of these things. I can't comment on 100 things every single week, all right? But I do definitely want to think about some of this stuff and try to
be cerumen or earwax buildup. If you get a hair in your canal.
I'm going to mute that because I'm pretty sure we don't want to hear that. Okay, that's all right. So there's all sorts of cool stuff. This one, as I just scrolled to it, I think that's really pretty interesting, talking about different types of bacteria, different shapes of bacteria, and what sorts of impact that might have. And I'm interested and a little curious about some of this. I don't know what that's. Let me dismiss that. There are definitely some potentials when it comes to shapes of things. And There are certain things where we've got receptors, these molecules fit like a lock and key and interact. There's a mention of coronavirus and some interactions with that as part of those spikes interact with respective host cells. When it comes to bacteria, there's definitely some potentials with that as well as we think about shapes of organisms. And there's comments in there about some of this as well, with potentials for maybe bacteria to be able to move or to not move. But when we think about like recognition, our body recognizes the bacteria not based off of its specific shape, like a rod or a coccyte, a ball or a big barrel type of shape. It's not really looking at that. And the spikes on viruses are very, very small that allow them to interact with receptors on cells. Bacteria are huge compared to viruses, all right? So it's a fragment or a fraction of that bacterial cell that our body can potentially recognize. I won't say it always recognizes, but possibly recognize. Specific fragments, little pieces of protein, amino acid sequences that give it a specific type of shape. And as we think of these bacteria, there's all sorts of other protein molecules that we would see on the surface. These things might be what's recognized. And there are these recognition proteins that microbes have on their surface that some of our immune cells can recognize and say, these suckers are foreign. This isn't our own stuff. It's not our own cells. This is something different, all right? So the shapes do have an impact, but it's not really the details of rods and cocci that our body could say, oh, that's a gram-negative rod, right? Or that's a gram-positive rod, or whatever. And you haven't been told what gram stuff is in the first place, but thin or thick cell-walled organisms. And that is also important when we think about some of that stuff, all right? I mentioned this one just a second ago with depression in the gut microbiome. There's all sorts of cool stuff thinking about our microbiome outside or inside and impacts that we have with that. And I think that's pretty interesting. I also, I looked at this one with maybe potential for dividers on those latrines. You know, the image that we showed with those Roman latrines and their sewage system, yes, there are potentials for disease to be spread, but really we were trying to show in that how advanced this society was and their ability to deal with wastes and pipe it away from the city to help minimize the potential for disease. I wasn't really thinking about, you know, sitting on the latrine next to somebody and potentially getting ill. But after I was reading through, I was like, oh, yeah, well, maybe there's something. And somebody had a comment about the communal butt wiper that I was like, what in the hell? I don't like the thought of that. Some sort of sponge that you could do your business with and then rinse it off and somebody else does their business. That's a little too much information. I felt like, holy cow, right? Who knew, right? Interesting, right? Somebody knew, somebody shared that tidbit, and that's something that I will now remember for forever, right? You can't forget something like that. All right. I liked this one, the bacteriophages to potentially treat stuff. Bacteriophages are viruses that target bacteria, and there is definitely some promise. There are some potential issues when we might think about some of these things and comments about how could we get mass amounts of virus. Microbiology helps us to do some of that. We can grow up huge amounts, truly. I'm curious, and I do think there is promise. There are also some potential issues when we do think about broad scale release of this, like antibiotics. Do we have the abilities to truly do that? With drug resistance, things like using a virus is a potential alternative. And there has definitely been some use with that. So, so many things, so many cool comments and questions and responses. I will tell you, when I was first thinking and being introduced to this, I thought, it seems pretty cool. But in the back of my mind, I wasn't exactly sure how this was going to work and the type of interest that would be sparked. And I do think it's good. I hope you guys have been able to get on here and look at it. And as we progress and it gets populated with more stuff, I think it will become smooth sailing. With that said, are there questions or concerns? Anybody tuning in from home have issues? Okay. Silence means no. Silence can mean whatever I want, right? Today, it means no. All right, I'm going to stop this. And like I said, I'll try to get a few comments in there, and I will definitely be looking at this in more detail as we progress along. Okay, so we didn't get too far, if I recall properly. I think we got about to here, a chunk of the way into the chapter. I assume we should be able to get through this today. I'm not going to show you other videos. Fortunately or unfortunately, I guess that depends on the viewer. Okay, let me get this. So we left off, we showed the Petri dish, we showed the inoculating loop. There was a question about the inoculating loop as well. Why do we heat that sucker up? Why do we need it in between each of the streakings to be heated up or to be heated in the first place? As we cook that loop, it sterilizes it. It ensures that anything that was possibly on it has been incinerated. It's been heated to temperatures that would destroy any sort of microbe that's on that. Now we would say it's sterile, right? And if you're doing the streaking on this plate, flaming in between these streaking motions ensures that we are killing what was on the loop, and then we start fresh with a new clean, basically slate. As that loop is pulled through, it helps to thin it out or dilute that specimen, so you get individual colonies instead of massive populations, right? That's why we flame the loop in between steps as well. All right, so Carlos Linnaeus, he wanted to give us a better understanding of what we were really thinking about as we try to understand and classify life. It was pretty common to just group things. If you move, you're an animal, and if you don't, you must be a plant, right? There's all sorts of things that are kind of in the fuzzy area as far as, is this a plant? Is it an animal? What is it, right? He wanted to get some better understandings and comes up with binomial nomenclature, the genus species idea, and helps us to better categorize and understand the world around us. So Haeckel, shortly after, wants to step it up a little bit and give us some more. So plants, animals, and protista, these single-celled oddities. A little bit later on, he adds another kingdom to this tree of life, the monarans. These are what we will call prokaryotes. It's our bacterial friends, all right? It's more than just bacteria, it's the archaea as well, all right? Microbes, all right? With that said, the protists are microbes as well, all right? Protista are eukaryotic, and the monarans are prokaryotic, so no nuclei, right? Or membrane-bound organelles, none of that stuff that we see in the top three, all right? So now we're talking 4 different kingdoms, right? So here we can see a progression, all right, with Linnaeus, animals, plants, and trying to give them names with this binomial nomenclature, trying to put some sort of name to a face, all right, and give us a little more solid understanding, some separations within that. Hegel separating it out further with the protists and then later the Monarans. And then as we shift a century later, Whitaker proposes fungi should be added into this mix. And we can see this tree of life getting more branches as we progress through. Few years after this, Carl Woese comes up with this idea that the Monarans, there's some crazy variation with these suckers, all right? We talked a little bit about this as we showed the Yellowstone hot pools. There are bacteria that can grow in some of these environments, and there are archaea that can grow in some of these environments as well, all right? And one of those questions was, bacteria are killed at these temperatures. These pools are hotter. It's archaea that are thriving at those above those temperature conditions, 100 and whatever, 170 or closer even to boiling temperatures. There are even some at the, deep down in the hydrothermal vents in the ocean where it's spewing out super hot temperatures through these vents where there are organisms that thrive. And they're also tolerant of massive amounts of pressure, which also helps them to maybe survive those super boiling temperatures. So Woese separates the Monarans, and this is where we see the different domains, the bacteria domain, the archaea domain, and eukarya, with the first two being prokaryotes and everything else up here, the eukaryotes, all right? And that's this, with this phylogenetic tree as we shift it over, because we can't fit it all right here. There's too much going on. So Woese and Fox, again, mid-70s, separate the bacteria and the archaea, which were once both Monarans, and now we treat them as separate. These archaea were defined as the extreme bacteria, these extremophiles, and now we've removed the bacteria. They are not exactly the same, all right? The archaea exist in all sorts of different habitats, all right? Some of these things we might think of as being very extreme. Others maybe not so much, all right? But we usually say these guys are extremophiles. They live in some sort of crazy adverse habitat. Super hot, super cold, acidic, basic, high pressures, all sorts of things that most would think of as being very difficult to live. These guys, one way or another, are able to eke out an existence, all right? So that's the idea with those guys, all right? There are all sorts of examples and images that they try to use to put things into perspective when we think about sizes of things. And you will get an opportunity to visualize some of these things in the lab pretty soon, all right? And I can tell you, when you first look at bacteria under the microscope, my gut tells me you will not be impressed, all right? They are very, very small, and you will see some that look kind of purple and some that look kind of pink. And you might say those look like little teeny circles, maybe little specks, and maybe they're a little bit elongated, and maybe you're like, I'm not quite certain, right? I think I see something, right? But I don't know. Is it a circle? Is it a rod? What am I seeing? They're very, very small, right? And they're microbes. That should make sense, right? They should be. So there's all sorts of ideas. And here we can see visible spectrum, and then we also start thinking microscopic range. You technically could be looking at a frog egg under the microscope, but you can see frog eggs with the naked eye, right? As we get smaller, a human egg or a pollen grain, you're not going to see these with the unaided eye, all right? And I will tell you, pollen compared to a bacterium, pollen is very large. It is awesome to see under a microscope. Looking at pollen grains and all of the crazy spikes on there. One look at that and I'm like, that makes sense why people have allergies. Something like that being inhaled, it's going to make you sneeze, right? And it potentially could stimulate another type of response. We'll talk about allergies and how that stuff happens a little later on. Some of you have them, and some of you probably don't, right? You could still get it, right? So plant and animal cells, these are all eukaryotic cells, and comparing it to a bacterium, they're monstrous in size, all right? About 10 times the size of a bacterial cell. And usually when you look at a eukaryotic cell, you get not as magnified as you would with the bacterium, and you still see huge features within that cell. For a bacterium to be visualized, you have to zoom in as high as you can with the magnification, and it's still unimpressive, all right? Just a little teeny speck, all right? Due to the super small size, all right? So in a eukaryotic cell with the microscope, usually at about 400 times magnification, you can see lots of details. The nucleus, maybe some organelles. Maybe in biology, you had the opportunity to look at some chromosomes as you were looking at onion root tips. And you could see those chromosomes, the contents of the nucleus that's spewed out and all lined up, right? That is super awesome, right? You don't see that stuff with bacteria, right? And they don't have, chromosomes that are all membrane bound. They've got a single chromosome, and it's really kind of just all over the place inside of the cell, right? So a red blood cell is our next one up here, and it's still large compared to these guys. The red blood cell's smaller than other normal, like our own body cells. It's also a little bit different as we look at this, kind of puckered up in here, looks sort of like a little donut. Our red blood cells are non-nucleated, right? There's no nucleus in there. They are different, and lacking that, it also means they're a little bit smaller, right? They're super flexible to get into all of our tissues as they are, moving through the capillaries. As they get into these spaces, they need to have that flexibility, and having all sorts of other structure inside would make them not as pliable, not as flexible, all right? So that also, the fact that they're lacking that makes them a little bit different when we think about some other details, like an immune response. They lack some specific proteins, and so our body kind of lets them go, right? There are issues with blood transfusion reactions, right? If you get the wrong blood type, but there are promises. The Red Cross says, thank you. It doesn't matter if you are type A and you give blood to a type A person, you can give them that blood and there won't be a reaction. And a lot of that is the fact that there's no nucleus and there's no proteins that are being expressed on the surface of this cell that says, I am me, right? It just says I'm type A. And that is why blood transfusions can work, because it's not an individual type of thing. That's when we start thinking about organ rejection with kidneys or liver or things like that, that's more of an issue because those are nucleated cells that do say, this is my liver, right? Or kidney or whatever, right? We'll hit some of that stuff in a bit. Now we finally hit our bacteria. These guys, super small, about a micron or two long, sometimes a little bit bigger, but not much, all right? Very small. And in order to visualize that, it requires, on a light microscope, the highest magnification. So it's magnified 1,000 times. And I said our animal cells, maybe about 400 times, and they look huge, all right? So if you get the animal cell under 1,000 power magnification, you are zoomed way in. And that's potentially when you start seeing the chromosomes in those root tips. I feel like you can see them at 400 power, but maybe not. Our little mitochondria up here, it's basically about the same size as our bacterial cell. We'll talk a little bit later about endosymbiosis theory and how mitochondria once were bacterial cells in the first place before they became the powerhouses of eukaryotic cells. As we move smaller, and this, I feel like we need a little bit of modification. Our light microscope's not getting us into this realm. You can't use a light microscope and see viruses. This is where we do our cutoff, the bacteria. You can't see a virus with a light microscope. Technically, there is a virus that is monstrous in size, and it could barely be visualized. But for all purposes in here, they're just too stinking small. You can't see them with a light microscope, all right? So this is where the electron microscope comes into play. And we can see different virus particles through the electron microscope. And really pretty impressive to visualize some of this. In the basement in the biology building, there's a big microscopy suite where there's all sorts of different microscopes. There is a transmission electron microscope that allows us to see surface material. This last semester, I had the opportunity, my first time, to actually see that microscope in action as we were looking at some bacteriophages, some of these bacterial viruses that were really very impressive. I know I've got an image saved somewhere. I will see if I can get one and show it to you guys. Pretty cool. I bet a couple of you in here might have it right on your phone. Okay. Proteins, lipids, other molecules, all of these things we rely on electron microscopes to visualize because they're too small. And then we get to the point of looking at things at like the atomic level. Regular electron microscopes don't give us those capabilities of zooming in that far. Close, but not quite. So we could see some of these bacterial shapes. We had a little hint of this in part of the PACDAC stuff that we were talking about before we got into lecture. Bacteria come in a variety of shapes and kind of sizes as well. They're all roughly about the same. And the way we're looking at them as singlets up here, but the way they behave also can be of importance, like how they are growing, how their population of them would appear. So here, a single one, a coccus. If we're talking multiple ones, we would say cocci. They're spherical or round, but they're three-dimensional. They're like big spheres, and they have potentials. Depends on the type of bacterium. We will see this in the next slide or two, where they may be single, they may be together like this. We would call this a diplococcus. They may be chains, they may be clustered. All right, we'll see some of that. The bacillus, these are the rod, the elongated shaped bacteria. We also call it a bacilli as the shape, but bacillus is also a genus. And with that, all of the bacillus bacteria, that genera, are all shaped like this, rod shaped, right? And there's numerous types of bacillus, like Bacillus anthracis, anthrax, right? Or Bacillus subtilis or cereus. Both of those bacilli are some kind of common food poisoning bacteria that you see out in the environment and potentially consumed and some illness with that. Vibrio are cholera, John Snow stuff that we talked about. It's shaped like this, kind of a comma shape. Rod but curved, all right? That's the Vibrio. Coccobacillus is kind of a mixture of these two, a sort of elongated cocci or a stumpy rod, depends on how you want to define it. Oval shaped, all right? And then the next two corkscrew looking guys, a spirillum and a spirochete. Basically, the spirochete is more twisted, more tightly coiled than the spirillum, but similar sort of characteristic. These guys move like an axial filament and kind of corkscrew or twist through their environment. Lots of the bacilli have flagella, and the arrangements of the flagella can be diagnostic. We're not going to get bogged down with that right now. But the bacilli can have some flagella that lends to motility. I believe our Vibrio can do the same. The cocci never have flagella. All right, we don't think of those guys being able to move the way that Bacillus or the spirochetes can. So we saw this before. So the archaea living in some of these crazy extreme environments set the stage for, at least in my boat, sparking all sorts of interest. I see this stuff and I am just inspired to wonder, what is it that's truly thriving in this sort of a habitat? There were definitely microbiologists that had similar questions. And a lot of our biotechnology, we really can thank researchers studying some of this stuff in Yellowstone to give us things like PCR reactions with Taq polymerase, the enzyme that's needed to replicate all of this DNA was isolated from a microbe in Yellowstone, right? And it ultimately changed all sorts of stuff as well, because the national park system was like, wait a minute, we feel robbed, right? They feel, and probably rightfully so, should be receiving some sort of kickbacks for this because it was isolated in the park, but that was before there was any sort of regulation like that, and now there is. You can't just roll into Yellowstone and collect some water and do some science with it, not without years of waiting and red tape and all sorts of issues, right? You know, you could be some yahoo that goes up there and throws a chicken in there to cook it up, but that's a different story, right? You probably heard about those guys. I think it was last year. They thought they could do that, right? We can cook chicken in here, right? You probably could, right? What do you think all those microbes think about? They probably aren't so happy about that, right? We will see. This image, I think, is also very impressive. Diatoms, they're a type of algae. There is all sorts of potentials with these guys, crazy shapes and sizes. They are things that we might think of as, you know, in a food chain, things that are consumed by all sorts of stuff. They are like the primary producers as we think about an aquatic type of situation. So like plankton, right? Similar idea, right? They also, the dead diatoms have kind of like a silica, kind of almost glass-like shell that is used in organic gardening. Diatomaceous earth is the dead carcasses of these guys that, when sprinkled around your tomatoes or other things, if an insect crawls through it, it gets into the exoskeleton or into the spiracles where they respire through their abdomen, and it basically cuts them to death, right? a brutal way to do pest control, but kind of awesome at the same time, right? Not if you're a grasshopper. Okay, this guy, oh, I could spend all day talking about Giardia. I won't bog you down with that, but it's a favorite. Giardia lamblia, technically it has a handful of names, and I don't know why, but in microbiology, we always say Giardia lamblia, and I don't know about like maybe Major's micro, but definitely in the intro micro, in the allied health micro, where we're thinking nursing stuff, we always spell it out as Giardia lamblia. And it kind of hurts my feelings the littlest bit as I teach parasitology and talk about these guys as Giardia duodenalis. That's who it really is. All right. But we can't call it that in here, and I don't know why, right? It makes no sense to me. All sorts of bacteria get different name changes, and they quickly change them. Textbook publishers put out a new edition because we just changed these names, right? We could have changed the name for this and sold you a new book a decade ago, but we haven't changed it, and I don't know why, right? Technically, they say Giardia lamblia, intestinalis, and duodenalis. All three of those names are accepted. It's the same player, right? That also makes me say, it's crap like that that makes people hate biology, right? When you give me three names for the same stinking thing, right? That's not fair, right? Do I need to know them all? Giardia lamblia, we'll call it that. But duodenalis is really what we are going with today. And that's where the bulk of these guys would be found in your duodenum, potentially taking up every single square centimeter or inch, depends on how you want to talk about it, all of the surface. Every single cell could be covered by the suckers on this little flagellate. This is a protist, right? And here you can see on the ventral side this huge sucker that basically sticks onto the cell like a suction cup, and there you go, all right? With the massive amount of surface area that they cover, you result in some nasty diarrhea, all right? You are pretty sure you're crapping yourself to death, all right? And you wonder why. And I imagine, oftentimes I blame Taco Bell for lots of things. And I would think, damn you, Taco Bell, right? What has happened, right? And it's not Taco Bell's fault. That's not what happened, right? Giardia is something that we get from the consumption of water. We drink the cyst. This is what we call a trophozoite, an active vegetative form. And then the cyst form with lots of protists are how we get infected. Some of them, it's the trophozoite stage, the moving around stage. Trophozoite's just a catch-all, right? Just the motile stage, right? So this guy lives in your intestines. You potentially are capable to release the cyst if you're not having diarrhea, all right? Then the cysts are formed, and that's what's infectious to the next culprit that happens to consume that cyst. If you are in a serious bout of infection with this, you're going to see a lot of diarrhea. And in those circumstances, it's the trophozoite that you see. And there can be mass quantities, right? There are instances where it has been described a patient presenting with some diarrhea and trying to figure this out. You collect it. A single diuretic stool from a patient has been found to have 14 billion parasites. A true buttload, right? That is awesome, right? And a bit unnerving. And like I said, you wonder, why am I crapping myself to death, right? This, all right? So we will go through, we'll talk about more details about Giardia and some of its potentials later on for sure, all right? It is pretty common parasite, and in Idaho, it's definitely our most common parasitic infection. Lots of people like the outdoors and potentially come in contact with fresh water where the cysts may be, and you accidentally consume that stuff. Typically, it takes about a month after the consumption of the water before you start feeling any ill effects, because these guys need to get in there and replicate and populate the GI tract. You probably consume a couple cysts, right? And once those cysts are consumed and hatch out, they start replicating by binary fission. They generate numerous new protists. And after about a month, you start feeling ill effects. By that time, you probably completely forgot about going camping and drinking some of the stream water, or potentially putting, frothy beverage in there. I've heard people put watermelon in the stream, let it chill, and then they slice it with a knife. And if you haven't washed that melon as you've cut it with a knife, you potentially put those cysts right onto the flesh of the melon. That's good stuff, right? As you know, I talk about all sorts of things in here. People are just like, holy cow, there's all sorts of nastiness with meat. You must be a vegetarian, right? But I just told you what you're going to get with your watermelon, right? You pretty much just can't eat anything. When I'm done with you, you'll just be like, what do I do, right? You're welcome, right? It's all fine and good, right? And with, Jon Snow's video, the take-home message is don't drink the water where you poop, right? There's all sorts of issues with that, right? More than just cholera. Giardia was one example. All right. This next image, these are some yeast cells. This is Candida albicans. It's a type of fungus, a yeast, a unicellular fungus. So they are culprits for all sorts of things. Vaginal yeast infections and oral thrush are two. They are the same, just different locations. Oftentimes with the thrush, it is something that we see in little kids. And as their immune system is first really kind of getting established, they sometimes deal with these types of yeast infections. With the vaginal yeast infection, we oftentimes think of that showing up as a result of taking broad spectrum antibiotics. And in the process of consuming these antibiotics, some of your normal flora gets decimated bacteria, and the fungus that is there in low levels and not causing any issues because the normal flora, your microbiome, is keeping it in check. When those populations get decimated, the yeast cells are able to proliferate, and that's when we start seeing these issues. So they oftentimes, and I think there was a comment on the pack back about some of this, probiotics like yogurt are definitely something that you should think about consuming if you are prescribed antibiotics, especially if it's a broad spectrum antibiotic, something that kind of is a kill 'em all sort of drug, sort of. Here we've got some more fungi, mold, on these oranges, and this gives us a macroscopic, something large enough to visualize with the naked eye. If we looked at these molds through a microscope, we would see all sorts of fuzz, all sorts of filamentous hairs or strands that we call hyphae. The big collection of it, all of it, like this whole orange, all of that fuzz on the orange, we would say is mycelium. Mycelium is the collection of hyphae. All of the threads makes the mycelium, all right? There are spores, the reproductive structures that are produced on the fungi, and that gives you kind of this sort of powdery white appearance with this type of mold. It's the spore that you're seeing. There's other fungi where maybe this is a different one, where it's more dark, more black, and spores that way. Penicillium, where we see penicillin, the antibiotic, is a greenish looking type of mold, and those spores especially kind of have a green tint to them. And usually it seems like that's what you see on a rotting orange, is kind of a greenish looking mold, most likely a type of penicillium, right? And we'll see it in a little bit as well. Oh man, more parasites, more stuff that I'm like, how much time do I have? I got a couple of worms up here that these guys, we're going to talk about them, but they're not really microbes. These suckers are massive or potentially massive, all right? The eggs or the cysts, dependent upon the type of parasite, those are microscopic, right? The juvenile or larval stages of some of these also microscopic, and in turn fit within the realm of microbiology, right? Even though these guys can be huge, all right? So here, the beef tapeworm, Taenia saginata, is the beef tapeworm infecting cattle and humans. The eggs, again, microscopic, but the adult worms, gigantic, all right? Between 4 to 10 meters in length, all right? That's a massive friend, right? So typically what happens in these circumstances, we harbor the adult worm within our intestine. The eggs are released. They get defecated out. If they happen to get released into the environment, cattle can consume this. As they eat the egg hatches, it bores through the stomach and gets into the musculature of the cow. It encysts, and now you have a cyst in the meat. You consume that meat, raw or poorly cooked, and that cyst, as it moves through your gut, the stomach acid causes it to basically hatch out, and the adult worm takes hold within your intestine, and it starts growing to the massive proportions. Awesome. The next one is dracunculus. Dracunculus mediensis causes dracunculiasis. It's called the guinea worm, right? And these guys, it's the consumption of water, contaminated water, that sets the stage for this. And in these circumstances, it's an aquatic invertebrate, a copepod, that has been infected with the larval stage of the worm. We consume that copepod as we drink it down and digest the copepod. The juvenile stage, that larval stage, is released. This worm takes a super long time to mature, almost a year after you've consumed it. It gets about 1 meter long, all right? The female will make, we're looking at, here's the heel, the Achilles tendon right here, and the ulcer where that worm has bored through. That female, when she's after about a year, she's been fertilized, she's full of eggs, and now she's ready to release. So an ulcer forms usually in the leg, and ulcer forms, and the first thing you probably want to do is put that in some water. You want to soak it. And that's what the worm wants you to do. As that happens, the juveniles get released out into the water to infect the copepod and to be consumed by you at a later date. Okay, guys, we will quit with that. I was so close. We almost finished it. We will pick up with this on Wednesday. Have a good weekend.
What do we need to do to prepare for the first lab?
Your first lab next week, you just need to go over the first bit of the material, like the safety stuff, first 6 or so pages of that, and then read through exercise 2.1, that's like ubiquity of organisms. You're going to do just a little bit of it, a little bit of swabbing. You're not going to do all 8 plates, just a small bit. So
Safety and exercise 2.
Yep.
Is that found in the textbook or individual?
In the lab manual,
Okay.
right?
Okay. Sounds good.